Hello everyone, my name is Kaelyn Fogelman and I am a PhD student at Auburn University. I am co-advised by Drs. Jim Stoeckel and Brian Helms. Today I will be talking about our comparison of Unionid feeding ecology between Lentic and Lotic systems. One of the barriers that we have in Unionid conservation is that we have a poor understanding of their feeding ecology. While we've elucidated the general constituents of their diets, we are still trying to determine where these components are coming from and how these animals are accessing them, either from benthic or suspended resources. Previous stable isotope studies historically have looked at planktonic material as food sources for unionids, but more recently we've had stable isotope studies that have looked at the contributions of benthic material, and we found that benthic materials can comprise a significant component of the mussel diet. We've also had studies that have shown that detritus or bacteria may make similar or greater contributions than algae depending on the study systems. And we've also seen that a lochthonous material can contribute more to the mussel diet than a tochthonous material. The objective of our study was to quantify food resources for unionids, and we do this by looking at their primary carbon source. So we wanted to compare food resources between Lotic and Lentic systems. The questions we asked were, between these systems, were mussels deriving carbon from benthic or suspended material? Was there variation across species in their dietary carbon source? And did we see differences in dietary carbon across system types? For our Lotic sites, we looked at 4 rivers in Texas. In the Llano River, we sampled Lampsilus bracteata and Cyclonius petrina. In the Navasota River, we sampled Cyclonius pustulosa. In the Colorado River, we again sampled petrina and pustulosa. And finally, in the Guadalupe River, we sampled Cyclonius neckii and Lampsilus bergmani. For our Lentic system, we looked at Gant Lake in southern Alabama, and we sampled 3 species there, Elliptio pullata, Buscania escambia, and Uterbacchiana hartfieldorum. At each of our sites, we sampled mussel foot tissue, fine particulate organic matter associated with benthic sediments, coarse particulate organic matter associated with decaying leaves or detritus, and suspended particulate organic matter isolated from the water column. Samples were processed by filtering, drying, encapsulated, and then being analyzed for stable carbon and nitrogen isotopes. The stable isotope data was then analyzed using Bayesian mix modeling with the Mixire package in R. What we found in the Lotic systems is that in the Llano River, our mussels' dietary carbon was primarily derived from sea palm, or that detrital material, with small secondary contributions from S palm, or a suspended material. In the Navasota River, pustulosa's dietary carbon was predominantly derived from sea palm, and we saw this consistently for our mussels in the Colorado and Guadalupe rivers. In our Lentic system, across all 3 species, our primary carbon source was F palm, or fine particulate organic matter associated with benthic sediments. In our Lotic systems, the dietary carbon contribution came from sea palm, or our decaying leaf material, and in our Lentic system, the dietary carbon was derived from F palm, or benthic sediments. So to answer our original questions, our animals were utilizing benthic material overall. We saw minimal species variation with a similar carbon source for all of our mussels in our Lotic system and another similar one for our Lentic mussels. And we did see variation across our system types. The shift in food source contributions from sea palm and Lotic systems to F palm in our Lentic systems could be an artifact of the riparian area to system volume ratio, where there is less overall allochthonous material available in the Lentic systems. The benthic contributions in our Lotic systems were expected as there is less algae available in a flowing system. But it was less expected that our mussels would utilize F palm instead of S palm in our Lentic system, and we are continuing to investigate the mechanisms behind this. I would like to thank my funding sources that allowed these projects to be possible, and all of the individuals and agencies that assisted with our fieldwork.